Let us resume our 37th lecture. So, in the previous lecture we talked about the movement of the mimosa pudica or the touch me not plant. So, that is one kind of movement where you touch the plant and it leaves curl or bent and the whole leaf droops. Here we will be talking about a second kind of movement, which is a very interesting movement where we talked about the carnivorous plants which eats insects and plants which respond upon touching of the insects. So, essentially it is the same thing. There has to be a pressure sensor or a touch sensor or something like that, what is in mimosa, but the mechanism is different from it. But the basic electrical activities are fairly the same. So, let us formally start this lecture with the plant bioelectricity of the plant movement in the Venus flytrap. Okay. So, this is basically what we are going to deal is the bioelectrical and biomechanical phenomena of carnivorous plant. The reason why I say bioelectrical and biomechanical, when we touch, there is a mechanical event followed by an electrical event where this leads to a generation of a series of ionic current or fluxes of ionic currents followed by a mechanical action. So, the way it works for those who are not aware of it. So, there are series of plants which have very diverse geometries than you have ever seen. These plants have certain alluring feature. Most of the insects, so we all know that the insects derive honey from the flowers and plants all over the place. This is all of us know. So, these plants look really very beautiful and the insects gets attracted either by the color or the smell or whatsoever. Okay. When they sit on these plants, they activate certain sensors and these plants are very unique. Their flowers are very unique. Say for example, you sit on it, it has a flap which will close or it will have something which will curl back like this. So, there are series of geometries by which and it can trap the insects, and today's lecture will be about that fascinating world of carnivorous plants and their movement, where we talk about how they are trapping the insects and certain intriguing phenomena and certain unsolved mysteries of nature. Okay. So, talking about it. So, some of you may have seen some of these pictures, some may not have seen. So, these are the examples, just handful of examples of a series of carnivorous plants which are available in nature. There is a huge population of it. There are at least 9 families of plants with 600 species distributed all over the world, and we really do not know how they evolved really. It is a very interesting story of evolution, why they evolved, how they evolved, what made these plants so very specialized as compared to other plants which just depends on water and other molecules to derive maximum part of their energy. So, if you look at the geometry of this plant in this slide from first, second. So, the first one, if you look at it, there is a hood. If you see the plant, so there is a cup like elongated cup like structure and there is a hood. Second one, if you see the same thing, but unlike a kind of it is much more more like ball shaped kind of. If you see the third one, if you see extreme right third one, you will see there as if there are a lot of you have seen the propeller or the A of a helicopter, it is almost like that. So, what happens from all the 4 sides or all the 5 sides or 6 1 2 3 4 5 6, all these 7 of them curl back like this. So, for example, they are all spread out like this. So, whenever they have to trap an insect, they all will become vertical and will close on it. Okay. Similarly, if you see the fourth one very carefully, so all those leaves which you are seeing spread out, they all will curl back. Similarly, on the 1 2 3 4 5 6 and in between, there is a small picture you will see where you will see some pointed between the 2 leaf flaps. There are kind of these pointed structures as if there are spines like this. So, this is a different kind of structure. Okay. So, in this lecture, we will be talking exclusively about this one family, but their mechanisms are same with barring aside few differences here and there. So, we will take one of these representative example and we will talk about their story. So, these are some of the among those 600 species. Either there will be pitfall traps of the pitcher plants, or the leaves folded into deep slippery pools. So, that is what I was trying to tell you. So, if you look at these plants very carefully, they really look very attractive. There is every reason for an insect to get attracted towards them. And not only that, if you see the inner part of these plants, inner part of these trap, you will see it is kind of a slimy or slippery kind of situation. Okay? So, if you again focus on this picture out here, the first picture, it is very slippery, and now if you follow this. So, the pitfall traps or the pitcher plants are leaves folded into deep slippery pools filled with digestive enzymes. So, they contain a series of enzymes which has the ability to digest digest a small insect, which plant does not produce unless otherwise it is needed. Plant depends on its own enzymes to break down the carbohydrates and likewise so on and so forth. But here these plants have a different kind of or added genetic machinery which allows it to produce series of digestive enzymes which could take care of these degrade the insect. Then you have the fly paper or the sticky or the adhesive traps of the sundews. So, they have a sticky material which exude out, causing sticky mucilage. Then you have the snap traps or steel traps of the Venus flytrap, what we are going to be talking about, water wheel plant and our hinged leaves. So, that is what I was trying to tell you. They have the hinged leaves with those sharp spiny structure. Then you have the suction trap, unique to bladderworts, are highly modified leaves in the shape of bladder with an hinged door lined with triggered hair. So, all of them have this hairy feature will be common. Then you have the lobster pot traps or the corkscrew plants are twisted tubular channels lined with hair and glands. So, if you look very carefully through all these structures, you will come across one common feature. They all have these hairy appendages or spiny structure which execute or which plays some significant role in this whole process of trapping the insect, digesting it and extracting out the nutrients. Okay. So, now if you see this picture in this slide, so most of the time the insects kind of comes and sit, getting kind of attracted about those mucilages, and that is where it gets trapped. So, we will talk a little bit more now. So, this is the trap we are going to talk about, the Venus flytrap or the insectivorous plant, the mechanism of movements and duration of the effects of stimulation in the leaves of Dinoea. Okay. So, this is how they look like. I told you that this small picture I was showing. So, look at the spiny structure of the leaves. So, they have two states, either they will remain open, as you could see in one of the leaves you see there is this red surface, that is the inner surface. Okay. So, near the crease where the two leaf jaws, so those are the jaws joints, there is a series of tiny hairs. You could see this hairy structure. If an unwary insect walks across these hairs, touching two or more of them in succession, this is very important, the leaf will close quickly enough to prevent its escape. Unable to escape between the hair-like teeth at the edges of the leaf, the helpless insect is slowly digested and absorbed by the leaf. Glands of the leaf surfaces secrete several digestive enzymes that help to decompose the insects. Once the insect has been digested sufficiently, the leaf reopens for another victim. So, now see this picture very, very carefully. You see the inner part of the leaf, the two leaves, rather it is one leaf, but it is just kind of curling with a hinge out there, which is red, and the outer part which is green, and then you see those doors, hinge doors on the edges. Okay. Now, there is something very interesting from this picture you cannot figure out on those red surface. I will be coming to that. Now, look at it very carefully. What you see on those red surfaces, you see this pointed hinge coming out, and they are very specific. They are not very many numbers. There, if you see in the first panel underneath which is written figure 3, you will see 1, 2, 3, 4 on one side, and probably 1, 2, 3 is visible to me out here. Oh no, 1, 2, 3, 4, 4 on the other side. Okay. And if you zoom on at each one of them, at least one of them, this is in the figure 4 what you see, as if there is a pin which is pointing out, a spiny structure, and this is how it looks like. Now, these structures are very essential. So, what happens is this, on these surfaces the insects get attracted either by the color or by the smell or by some slimy sticky fluid or something, some fluid or whatsoever is present in between those, that leaf like thing, it looks like this. So, the insect comes, as if the insect comes out here. Now, imagine this pen as the insect. Okay. Now, one of the previous slide I was telling you that insect has to remain there for a certain amount of time. Now, these structures what you see, 4 on one side. Now, if you concentrate on the slide again, there are 4. So, these are the sensor elements. So, now, if I go back, I was telling you this, sorry. Near the crease where the two leaf jaws join, there is a series of tiny hairs. Okay? If an unwary insect walk across these hairs, touching 2 or more of them in succession, the leaf will close quickly enough to prevent its escape. This line is important. Now, what does that mean is, these spiny structure what you see, the 4 in one side, 4 in other side, the insect has to touch 2 of them in succession. If it does not do, then the leaf will not close, as if 2 of them leads to a cascade of reaction. Now, if you see it, this is how it looks like. When the insect touches 2 of them, then the leaf closes and it prevents its escape. So, this is very important that you understand this part. So, now see the sequence of events, how this is taking place. 0 second. Now, this has been done without a insect. How it has been done? So, these experiments could be done in several ways. So, how we discovered that it has to touch those 2, why, from where this line is coming. If an unwary insect walks across these hairs, touching 2 or more of them in succession, the leaf will close quickly enough to prevent its escape. So, the way they figured it out is very interesting. So, now let us come back to this picture. Now, you take a mechanical probe. You see this picture? You take a mechanical probe and you start touching those 4 on one side, 4 on the other side, one after the other in succession. But there is a very tricky time delay. Now, if I show you on the board, it looks like this. Say for example, this is, if this is the inner surface with those spiny structures coming out. So, I take a mechanical probe like this, touch it, followed by the second one. This is the first touch, then this is the second touch. But this first and the second touch has to be done in such a way that there is very limited, like if between touch 1 and touch 2, there is a huge gap of time, then this phenomena won't be seen. You understand? This phenomena can only be seen if there is an optimal time between T1, touch 1 followed by touch 2. If this does not happen, that optimal time window, then you won't be able to see this closure phenomena. And that is what it meant when the insect comes, it has to touch this and this, or this and this, or this and this in succession with a very limited time delay. If that does not take place, you won't be able to see this process. Now, if you follow this with this picture on the board, or now if you follow this picture on the slide, that will make more sense. So, this is what you see out here. There is a mechanical probe which is touching those 4 and 4 hair like structure, 0 second, 35 millisecond, 67 millisecond, likewise up to 64 seconds. And in that whole process, by inserting an electrode, so the way it has been done, these are the outer surface, you insert electrodes like this, and you can do the recording from an eigenamplifier about how the electrical activities are taking place when you are touching. So, what you see essentially out here, there is an electrode which is inserted here, and there is a mechanical stimulator which is ensuring that you are disturbing this surface. Okay? This is how it works. You have to touch them mechanically, these sensor elements, and you have to do the electrical recording simultaneously. So, you touch it, electrical recording, you touch it. So, if you plot the graph like this, it should be like this. So, this is for the mechanical stimulus. So, stimulus followed by an electrical signal, stimulus followed by an electrical signal with a small delay out there. This is how it works. So, this is exactly what this picture is trying to show you, that 0 second, 33 millisecond, likewise up to 64 second, and on 64 seconds you will see the leaf has closed. And by trial and error of doing these things, people had figured out how this Venus flytrap works. Okay? Now, this is the kind of response properties of sensory hairs excised from the Venus flytrap. So, those what you see are called the trigger hairs. So, these are those trigger hairs, what I am showing in the board. When these trigger hairs are triggered, you see the closing motion of those along the hinge, along the Venus flytrap. Okay. Those trigger hairs are in a certain angle, and over 100 millisecond time, that angle changes likewise. Okay. And you could see, if you see the slide, you will be able to figure that out much better. Okay. So, this is how that movement takes place. So, trap closure and trap closure memory. Trap closure is a precisely controlled process. This is not a random process. This is very, very tightly controlled process. As I was trying to tell you, as I was showing you, that you given a stimulus with a delay, you will see electrical response. So, there is an optimal time window between these two. And that optimal time window discovery was one of the very interesting feat for those people who are working in this area. Okay. So, coming back, how these studies are being done, first of all, in order to understand it. So, the way these studies are being done is something like this. So, you see the slide picture, and now I will show you. So, if this is a Venus flytrap situation with, okay, all those spiny structures coming, and these are those specific cells. Now, you remove it like this. You remove a specific spine like this. Okay. Now, what do? You insert an electrode out here. You can keep it in. And you have a mechanical sensor which is hitting on it. Mechanical stimulator. And this is your electrode. Now, this is what I was trying to show you in the slide. If you see the slide, this is exactly what it is. You have an electrode which is inserted through the center, and you have the mechanical stimulator which is hitting upon it. So, whenever you are hitting upon it, what you are doing, you are measuring the electrical activity. So, if you read through this excised hair preparation, you see the excised sensory hair was supported by an electron microscopic grid. So, the electron microscopic grid, what you see out here in this picture is this. You see this electron microscopic grid. So, this is the electron microscopic grid, and if you follow this, and was insulated to isolate the upper podium chamber from the lower chamber. U1 defines the potential of an electrode in contact with the bathing solution. So, you are measuring with respect to the bathing solution, and then you have this mechanical stimulator coming through. This is how most of these electrical activities of these kind of sensor elements are being discovered over the period of time. Okay. So, now moving on to the next slide, if you look at this structure very carefully, this is the cross section and the longitudinal section of the hair cells. So, these are called hair cells. Please do not mistake, like you have hair cells in the ear also, very similar to that though, but these are the hair cells of the plants. Okay. But the functionally they are same. Both of them are mechanosensitive structures. One is in plant, the other one is in the ear. Okay. Now, the cross section and the longitudinal section of the hair cells. The cross section at the left was selected from an indented level of a hair. Indented sensory cells form the outer margin. So, basically if you see this structure is something like this. So, they have on the periphery you have the sensor elements, that is where your this kind of touch sensors are acting, okay, on the surface. So, these are those sensory cells, what you see out here in that structure. And if you follow the slide now, the longitudinal section at the right, the smaller cells occupy the central region of the tissue, and the longitudinal section at the right includes only a fraction of the total liver tissue. Okay. From the top and bottom, the longitudinal section includes woody liver tissue, the podium region. So, there are certain parts which are non-living part, and they are existing out there. Okay. Now, from here if you look at it, you can stimulate them at different typical results of destructive dissection. You can really do a series of recordings based on how much stimulation or mechanical stimulation you are giving over a period of time. Okay. You guys can go through this. This is a very interesting thing, but what I will be more interested is to develop how people have developed this algorithm out there. The electrical signaling in venous flytrap induced by a piece of gelatin. So, gelatin is acting as the stimulating one trigger at here on each lobe. So, one AgCl electrode was located in the midrib, and the another AgCl electrode is located in the center of the lobe. Okay. The channel 1 shows the solitary wave between the lobe and the midrib, and the channel 2 show the electrical spike in the lower part of the leaf. Okay. So, this is how these recordings. So, if you go back to this recording or even this is how it is pretty much the same assembly how these recordings are being done. Okay. Now, coming back, what exactly is happening if we really look at it very carefully. So, this is an open trap. Now, follow it very carefully. Now, there is a prey. So, this is an open trap. Now, there is a prey which came here. This is the prey. It touched this one. So, this generates a receptor potential RP1. Now, see the slide, receptor potential, the first mechanical stimulus, followed by it puts its legs on this one and generates another receptor potential which we are calling as RP2. This is exactly the same thing. Now, if you follow the slide, this is exactly the same thing what happens when the light falls on your rod and cones or the sound wave hits upon the hair cells of the ear likewise. Okay. It's the same thing. Okay. So, there is a change in the receptor potentials followed by uncouplers or ion channels blocker. They get activated, and as soon as those ion channel blockers gets activated, this leads to two different action potentials. So, one action potential is coming from RP1, the other action potential is generated from the other one where it is hitting. So, one action potential out here, another action potential out here, AP2, and you have AP1. So, now there are two action potentials which are generated. Now, these two action potentials, if you follow the slide now, this leads to the electrical charge transduction and charge accumulation in the ATP and hydrolysis in the midrib. Now, followed by the next level. So, the part 1, if you follow this slide very carefully, you see there are two dotted lines. First one is the sensory activity. Part 2 is the electrical activity. Now comes the part 3. Part 3 is your mechanical activity. So, look through the trap closure in 0.3 seconds. Okay. Two holes in the meshworks allows the small prey to escape. So, there are situations when, say for example, these two are folding and the insect is really agile and left. What will happen? How this algorithm will work? Coming back to the slide, if you follow the left hand, the holes in the meshwork allows the small prey to escape. If that happens, this system senses it, and within 1 or 2 days it will open. Otherwise, if the trap closes in 0.3 seconds and the insect fails to escape from it, then the prey is captured. This is followed by, now on these surfaces, there are a lot of digestive enzymes which are synthesizing enzymes which are present there. So, if this one closes, so now the trap becomes, one second. First you have the sensory activity where your insect has touched on those hair cells, followed by a change in the receptor potential 1, receptor potential 2, leading to an action potential AP1, AP2, and this whole thing is electrical activity. Okay. Now, followed by a mechanical action. Now, this mechanical action is in the form of this is the open situation and here is closed situation. As soon as this one closes, there is a lot of secretion of from the surrounding cells out here, lot of secretion of digestive enzymes. The reason why this whole mechanism depends on two receptor potential changes or two action potential changes probably is nature is very conservative. It does not allow the mistakes to take place. So, there may be a deflection or something, but the insect may just run away or something else. So, in order to ensure that there is it must have been an adaptive process, and the time difference between the two, that is very critical. From receptor potential 1, receptor potential 2, if there is this time window goes fairly high, then this whole process is going to cancelled out. It's not going to take place. So, this is very important that this takes place very fast. I mean, there is an optimal time window. Without that optimal time window, this cannot be executed. This is very, very, very clear. Okay. Now, if we look at the different plant memories, what we are having today in the world. So, electrical signaling in the memory play a fundamental role in plant responses. So, one of the thing is the most foremost one is the storage and recall function in the seedlings when the seed germinates. This is a classic example of it has certain degree of memory, it remembers it. Next is the chromatin remodeling in the plant development. So, these are overall. Then you have the transgeneration memory of stress. They could respond to the stress as if they have already memory recall switch by which they know how to respond. Then you have the fourth one as the immunological memory of tobacco plants and mountain birches. Okay. I wish you students please go through them, go to Google and give searches and see what all these significant, because we are only talking about the electrical phenomena, but there are different memory events which are taking place in plants. Okay. Now, then on the fifth one, if you come on the slide, vernalization and the epigenetic memory of winter. Okay. Winter hardness and cold hardness and all those things. Induced resistance, the sixth one, and susceptibility to herbivores. Then you have the seventh one, the memory response of abscisic acid entrained plants. Okay. Then you have the eighth one as the phototropically and gravitropically induced memory in maize. And the ninth one, ozone sensitivity of grapevine as a memory effect in the perennial crop of plants. Then the 10th one, which we have talked about, the memory of a stimulus. This is exactly what we talked about. This is called the memory of the stimulus we have talked about. Then you have the 11th one, you see in the slide, systematic acquired resistance in plants exposed to pathogen. Over a period of time, they act on it. Then you have the electrical memory in the venous flytrap, 12th one, what we have talked here. Okay. So, the way the electrodes are being, so here I will be discussing with some of the mechanism how it is being done. So, you see the veins out there, and you see the trigger hairs which are present there in the shadow EA. Now, using charge injection method, it was evident that the application of an electrical stimulus. So, this is very interesting to note. So, as of now, we talked about that there has to be a mechanical stimulus. Okay. There has to be a mechanical stimulus before this whole process takes place. Now, it was being observed that whenever there is a change in the receptor potential out here, what receptor potential change means? That means those tissue are sitting at a resting membrane potential, which is shown by RMP. Okay. Now, by some mechanical stimulus on them, there is a change in RMP, resting membrane potential, and that is what we are talking about, resting potential change. Fine. That is what, if you remember, sorry, the receptor potential change. Now, come back to this slide. Now, if I could go on the third line, open trap, prey, and the receptor potential. So, there is a mechanical stimulation leading to receptor potential change. But think of it, without giving mechanical stimulus, if electrically I induced a receptor potential change, I change the potential electrically, will this whole process be executed? Now, coming back to the slide, just imagine if you see this line, mechanical, the first mechanical stimulation, second mechanical stimulation, what I was trying to show you, the mechanical stimulation taking place here, mechanical stimulation taking place here, likewise in the RP1, RP2, you see the mechanical stimulation here, mechanical stimulation here. I do not give the mechanical stimulation. Okay. What I did is I changed the receptor potential by an electrical method. Will still this whole cascade of event, coming back to the slides, if you look at it, will this whole cascade of event takes place, and that is the experiment which was being done. Using charge injection method, you can actually change the potential of the receptor. Receptor potentials could be changed by charge injection. Using charge injection method, it was evident that application of an electrical stimulus between the midrib positive potential and the lobe negative potential causes venous flytrap to close without, mark this line, without any mechanical stimulus. Stimulation pulse voltage is sufficient for rapid closure of the venous flytrap was 1.5 volt. Okay. This is very interesting to note that this process is in a natural condition. This process is influenced by the mechanical response obtained by the plant because of insect sits on top of it, but you actually can orchestrate the whole process of the closure without the mechanical stimulus just by inserting an electrode here, and injecting current to change the receptor potential. If you change the receptor potential, this whole cascade of event will follow without doubt. So, what is the take home? And here in the slide, if you come back to the slide, you will see this is how these kind of charge injection methods are being followed, being used precisely to estimate the amount of electrical energy necessary to cause the. So, what you are essentially doing, so there is a mechanical energy here. So, now you are replacing the mechanical touch sensation with an electrical stimulus. So, when we are talking about the electrical stimulus, essentially what you are telling is we are injecting a specific amount of charge particles. Okay. So, this is what the charge injection methods will highlight, has been used precisely to estimate the amount of electrical energy necessary to cause the closing of the leaves. The two critical parameters have been analyzed, the amount of charge and the applied voltage. Both parameters are tested to determine the minimum amount of charge and minimum voltage sufficient to close the plant trap. The double pole double throw DP/DT switch was used to connect the known capacitor to a voltage source during charging and then to the plant during plant stimulation. So, this is how this whole thing is being carried out to quantify the charge and quantify the charging voltage, and this is how this whole thing is being followed. So, you see there are on off at the two levels. Okay. So, I wish you guys kind of go through them in a small, large, depending on how much charge you are giving, the closure mechanism changes its slope. Okay. So, now if you see the slide, open trap, first stimulation, second stimulation, wait for mechanical stimulation by the prey, change in the receptor potential, the sensory memory, action potential, which is a short term memory, followed by ATP hydrolysis, which is basically a biochemical phenomena, followed by a proton transport, fast H plus transport, water transport through aquaporins, which are the water channels, fast trap closure. So, small prey, if the holes in the cilia mesh allows the small prey to escape, the trap reopens after 2 days. But if that is not the case, and if the insect is completely trapped, and with the effect of the digestive enzymes which are secreted by the leaf surface, the cilia mesh is locked. Looking at the slide, if you realize, then it will not open before 4 or 5 days. Okay. So, this is the overall schematics which could be done, and you can execute the whole process at the line 2 of the slide by electrical stimulation, by giving it charge. Okay. So, there are several models which are involved in this. One of them is called, in the slide you see, hydroelastic curvature model. I wish you people to go through them. Basically, it says that after receptor potential, action potential is generated, which are propagated to the midrib of the plant through the plasmodesmata. So, these are the connections. If you remember in lecture 36, I showed you where the plasmodesmata lies, and the connection between the xylem and phloem and all those things. Okay. So, those plasmodesmatas are very important. So, please go through the lecture 36 to see. I gave you in between an schematics of the plasmodesmata. Okay. Now, following on the slide, the action potential can be inhibited by uncouplers. So, you can use different kind of potassium channel blockers, and you can stop this action potential. So, you can actually stop this whole process here itself. You may not allow this to proceed further. Now, coming back to the third, the venous flytrap memorizes the first electrical signal for a short period of time, and as soon as the second action potential propagates into the midrib in 1 to 40 seconds. So, this is the time window, electrical signal. So, between this and this has to take place between 1 to 40 seconds. That is the critical time window, what I was telling you, the T between T1 and T2, or some mechanical stimulation at 1, mechanical stimulation at 2. If there is 1 to 40 seconds delay, then this process of ATP hydrolysis and everything is going to take place. If you see the slide, that is the third box is going to tell you about that. Okay. If you follow it, the venous flytrap, 1 to 40 seconds, electrical signals activate the ATP hydrolysis, starting fast proton transport initiation. So, basically the whole proton transport, when we are talking about out here, this place becomes very, very acidic, because there is a huge amount of protons which are moving out there. H plus ion concentration is really gearing up. The fast proton transport induces transport of water and change in the turgor pressure. The leaves of the venous flytrap actively employs turgor pressure and hydrodynamic flow for the fast movement and catching insect. In these processes, the upper and the lower surface of the leaf behaves quite differently. This is exactly the same situation as you saw in Mimosa pudica. During the trap closure, the loss of turgor by parenchyma lying beneath the upper epidermis, accompanied by the active expansion of the tissue of the lower layers of parenchyma near the under epidermis, closes the trap. The cells on the inner face of the trap jettison their cargoes of water, shrink and allow the trap lobe to fold over. The cells of the lower epidermis expand rapidly, folding the trap lobe over. The minimum elastic energy of the leaf, including mean and Gaussian curvature, corresponds to the closed state. So, this is what I was telling you about the hydroelastic curvature model. So, there are several other models which are involved in this whole process. Please go through them. And so, this is the overall what I wanted, that there are mechanical events, just like what happens in the nervous system. There are mechanical events which are translated into electrical event, and the mechanical event could be simulated by inducing charges into those tissues, and that is exactly what this experiment was all about. So, one second, yeah, this is the one which is all about, where you are injecting the charges into it. So, overall if you look at it, so by the action and reaction, it looks very fairly similar, that what is there in the animal system is pretty much followed by the plant world. But unlike the animal world, plant do not have a very specialized nervous system, well demarcated nervous system. And that is why back in 1926, so J.C. Bose came with his idea that nervous system of plant are rudimentary, or the probably the very the kind of the most underdeveloped nervous system before, as the animal evolved, they developed much more higher organized structure. Okay, so I will close in here, and I will try to give few other handout which may help you to appreciate this whole process.